I feel like it would be sacrilegious to talk about muscle contraction without talking about Arnold, right? True story, I actually met Arnold Schwarzenegger and had a chance to walk with him through some of my lab space at a previous institution. Really great guy. He's a lot smaller in person than you would think, but I think he's probably back on the sauce because he's getting big again. So how does muscle contraction actually work? We just talked in the last video about what are some of the contractile proteins, what are some of the specific aspects of a muscle cell that differentiate a muscle cell versus, say, a skin cell or, say, a vascular tissue cell. Well, the primary way that a muscle is going to contract is with those contractile proteins. And that makes sense because that's the name, right? Name follows function, form follows function, whatever. Essentially, we have this current best working hypothesis or model for how muscle contracts. Does it mean that it is infallible? No. Does it mean that it is without a shadow of a doubt as true as it could possibly be and we can label that sucker a theory because we know it's the truth? No, not necessarily. But this is our current best guess at what's going on inside of the muscle when it's contracting. Essentially, that's going to be what's called the sliding filament model. Imagine actin and myosin as filaments. That's instead of strands, consider using the term filament. Essentially, what's going on with the sliding filament model is that you're having a contraction because actin is sliding over the top of myosin. That myosin is actually going to be held in place, and you're going to be pulling that actin across the top of it. If you remember, you've got Z discs on both sides that are anchoring that actin. So if you pull that actin over top and keep pulling, imagine it's like hand over hand rope pulling, you're actually going to be pulling those Z discs closer and closer and closer together. That's going to reduce the overall muscle length, so it's going to contract the muscle. Where are we getting the energy to even contract a muscle? Imagine how much energy it would possibly take to move or translate the forearm in a different or in a plane. Imagine how much effort it would take to translate the forearm, create movement at the elbow with elbow flexion. Now add 200 pounds to it. Well, that's excessive. Add a 25-pound dumbbell to that. And imagine how much more ATP you would have to have in order to fuel the contraction. Well, essentially, the energy from the contraction comes all the way down to ATP. It's why we spent so much time talking about ATP and why we spent so much time talking about these energy systems and bioenergetics is because this is where we're going to actually be producing that mechanical work. This is where we're going to be using that mechanical energy is muscular contraction, whether it's coming from our skeletal muscle or cardiac muscle or smooth muscle. This is where it's getting used, a large spot of where that ATP is getting used. So essentially, we're using that ATP because on the myosin, there's actually these two globular heads. So it's like a stalk and then two globular heads. And sitting on each one of those globular heads is going to be ATP ACE. If you remember, ACE means enzyme, and what ATP ACE is responsible for is hydrolyzing the bond or breaking the bond on ATP to make ADP plus anorganic phosphate. So there's a liberation of energy that's going to be occurring. For every single myosin head with ATP to be completely fueled with ATP, and we had a full contraction of every single myosin head, we would only move that muscle 1%. We would only have a 1% shortening of that muscle if every single interaction occurred and we had every single ATP molecule get broken down and everything worked flawlessly. We would only move the muscle 1%. That's why these energy systems have to keep running. That's why we keep having to have fast energy and long-term energy availability is to run through repeated skeletal muscle contractions. So essentially, the moral of the story here is that we need a lot of ATP in order to fuel contraction. It's why everything that you learned in XPhys is important for those 4460 students, and that's why we have skipped around chapters quite a bit this semester already in XPhys, is to get you guys the baseline knowledge that you need before we can dive too much further. Like, awesome, we get this concept of sliding filament model at 30,000 feet. How do we actually integrate signals? How are we going to take a signal from the dome and put that all the way into the muscle? Well, this is where excitation contraction coupling really comes in. And this is where we start really taking advantage of that neuromuscular junction, because if you remember, our bodies love electricity. Our bodies really run on electricity. So that signal that's being sent from the brain all the way down the axon, like down neurons to the spinal cord, out the axon, that the cell body sitting inside the spine, the axon runs all the way out to whatever motor unit or end plate it's trying to reach to. You're going to have this electrical signal, and then that muscle is going to actually receive neurotransmitter signal and convert that signal into electrical signal to create skeletal muscle contraction. So excitation refers to the activation of the muscle, or the activation and the sending of a message, whereas force production is the contraction side of things. So it's signal integration, and then also how do we contract the muscle as well. Starting from the top, we're going to have our excitation phase. So our excitation phase for muscular contraction is really going to start when the signal from the brain is received or reaches the neuromuscular junction on the presynaptic side. So this is at the neuron side of that synaptic cleft. So you're going to have electricity signal that gets sent down. You're going to have these presynaptic vesicles fuse with the neuron membrane. Sorry, I'm knocking on the desk because I'm trying to. You're going to have these vesicles fusing with that cell membrane, releasing neurotransmitters into the synaptic cleft. Those neurotransmitters are going to span that synaptic cleft. They're going to kind of float around in there, and they're going to get bound to receptors that are on the motor end plate. If you remember, the motor end plate is the muscle side of that neuromuscular junction. Once you receive a certain level of signal from the muscle, you're going to hit a depolarization. I'm trying to creep up because my mic keeps... Once you reach a certain level of signaling, you're going to actually depolarize the muscle cell. Just like you depolarize the neuron, you're going to depolarize the muscle cell. When you depolarize the muscle cell, it's going to travel deep through the muscle because of those T tubules, because of the transverse tubules, and because of their structure. You're actually going to send that signal all the way through. So every single muscle fiber in that motor unit is going to be hit by the same signal. As soon as that signal runs through the T tubules and passes next to the sarcoplasmic reticulum, that electricity, that voltage, is literally going to open the floodgates, if you will, and allow calcium to leave the sarcoplasmic reticulum, or more specifically the terminal cisternae, which are these sacs that sit within the SR right next to the T tubules. You're going to flood the system with calcium, right? And that's exactly what you see here. So you're going to have a neural impulse traveling down an axon, synaptic vesicles fusing with the membrane, releasing neurotransmitters. Those neurotransmitters are going to bind to the receptor, and that binding to the receptor is going to cause a depolarization of the muscle cell. It's exactly, if you remember, what's going on here. So as we move more positive, that means that we're going away from polarity and towards a dipolar state, or a non-polarized state. We're going to have an action potential get sent. Remember, we have this all or none principle where once we hit a certain threshold, boom, we're having an action potential. We're going to repolarize, and we're going to be back to normal. Everything that we went over with neurons is happening here with muscle cells as well. So we know how to send the signal, and we know how the muscle receives the signal. How do we then take and interpret that signal and put that signal in to contraction? How do we actually make the muscle go, right? Well, that calcium that was released from the sarcoplasmic reticulum, or lateral sacs, or terminal cisternae, because of the T tubules, because of the electricity, is what's going to really initiate the contraction phase. So the calcium that's released from the sarcoplasmic reticulum is actually going to get into the muscle cells specifically, and it's going to bind with a protein called troponin. Remember, troponin is located on actin and is actually on tropomyosin. So when you bind calcium to troponin, you actually shift the tropomyosin shape, right? So tropomyosin would normally be blocking the ability of myosin to grab actin. But when you bind calcium to troponin, tropomyosin will end up rotating away, and it will actually allow for myosin heads, those globular heads with ATP on them, to bind to the actin, right? So it'd be like finally being able to put two pieces of Velcro together, or hook and loop together, so that you can actually create a bond, if you will, create a molecular bond. What I mean when I say a cross bridge is that it's where the myosin heads, it's where these globular heads are actually able to connect. They're actually able to click onto that actin and create a bond. So they're actually able to join together. Myosin and actin are now joined, and it's because of this concept called a cross bridge. In order to get a movement to occur, we actually have to drop off that inorganic phosphate, so that third phosphate on the ATP. We actually have to drop that off, and we have to drop off the ADP in order to get that full power stroke. And that full power stroke is going to be what actually causes the ratcheting motion over of actin, or the sliding motion over of actin, right? Myosin, actin, cross bridge, PI drops off, ADP drops off, and we're actually pulling that actin across. In order to get this to release, we actually have to bind an ATP back onto the myosin head. So when we bind an ATP back on, we break that connection, we break that cross bridge, and when we hydrolyze that ATP, when we break the ATP down in ADP plus inorganic phosphate, we recock that head, and we're ready for another cycle, and we're ready for another cycle, and we're ready for another cycle. So that's what's going on with these steps. So if we start up here where it says none other than start, we're going to see that calcium is released from the lateral sacs, from the sarcoplasmic reticulum, from the terminal cisternae, whatever you want to call it. That calcium is going to travel deep into the muscle, and it's going to actually bind with troponin and allow for there to be active myosin binding sites presented on the actin itself. When those sites get revealed, you're going to see that the myosin head binds to the actin. In order to create the power stroke, in order to start generating the power stroke, you're going to drop the inorganic phosphate, right? You can see here in step 3 that we're going to actually release the inorganic phosphate to initiate the power stroke. And then as we continue on that power stroke, we're going to allow for the sliding of actin down and across myosin. We're going to have a release of ADP, allowing ATP the chance to bind myosin again. When myosin and ATP bind, you're going to have a breaking of myosin, a breaking of the heads of myosin away from actin, which means that you're no longer in a cross bridge state. So there is no connection between these two anymore. Once that ATP gets hydrolyzed, it kind of cocks all the way back so that it can be ready to bind again. And there's going to be a need for more calcium. So if calcium continues to stay in the system, we're going to continually have active binding sites for myosin and actin. If calcium leaves the system, this is now done. There's no more contraction occurring. If calcium is there, we're going to make sure that we keep going through this system. We're going to have a dropping of inorganic phosphate, a dropping of ADP, a binding of ATP, a recock, power, right? Like all those steps are going to continue as long as calcium remains the same. However, if calcium disappears or is sequestered, we're not going to go through this anymore. We're not going to have contraction. And as we pull those actin filaments across the top and we're moving those Z discs, if we have no more calcium, if we have no more ATP, we're not going to continue to move, because without ATP, we can't break the bond away from actin and myosin. And without calcium, we don't have any active binding sites any longer. How do we relax the muscle? If these slides were all on contraction, how do we actually relax? Well, two different ways. One, we need nervous system input. We need signal to input in order to cause a contraction. There needs to be a lack of signal or an inhibitory signal to actually inhibit or stop the contraction from occurring. When there's a lack of an end plate potential, when there's a lack of a depolarization of the muscle cell, we're not going to have a release of calcium, And without the release of calcium, our bodies are going to just sequester that calcium and put it back where it belongs in the lateral sacs, terminal cisternae, sarcoplasmic reticulum, whatever you want to call it. It's actually the removal of that tropomyosin, or the removal of calcium from troponin, that reforms the tropomyosin block and does not allow myosin cross bridges anymore. So that's essentially how muscle relaxes. We'll go through this state of contraction, and when we repeal nervous system input and pull back calcium, we're no longer able to contract, so the muscle relaxes. And that's what this whole slide is going to be putting everything together for you. So steps 1 all the way through 7 is going to be highlighting what's going on, or I guess 1 through 9. Sorry, it starts up here at 1 and it travels all the way through 9. Is going to be going through excitation, through contraction, and eventually to relaxation, because we no longer have the input from the nervous system as well as the calcium.